The Xerra is a new tool from Emit Imaging for Cryo-Fluorescence Tomography, or CFT. CFT is a powerful molecular imaging technique which can be used for difficult-to-study cancer pathology, such as ovarian cancer models. In this study, HER2-expressing tumor cells, which co-express TD tomato, were injected into the abdomen of a mouse. After tumor propagation, a novel probe with HER2 protein targeting antibody, labeled with Elexaflor 647, was injected into the tail vein of the animal. The lab was previously only able to image these animals with an IVUS. This IVUS data shows that there is some correlation between the probe and tumor, but there is no ability to ascertain how much nonspecific binding there is and how much tumor is not labeled. We see that there's probably renal clearance, but do not know how much that contributes to the abdominal signal. Also, the data would be very different depending on the position of the animal, so longitudinal studies are not good. It is also very difficult to perform histology on the abdomen of an animal, as it's a very large slide area that most microscopy equipment cannot handle. Once the lab installed a Xerra, they could still run the IVUS data over time and then take the entire animal to produce a full 3D CFT dataset of both the tumor cells and the probe. The Xerra gives us a full view of how much probe is hitting the tumor or not, and how much tumor was and was not labeled with the probe. The CFT also shows us that the renal clearance is very low, and there's a considerable amount of tumor that is labeled. This was investigated at several time points to better understand their probe kinetics. Each voxel of this dataset was a ratio of the amount of tumor and probe and their correlation. This showed the researchers that the probe was targeting the tumor very well and that there was very little nonspecific binding outside the tumor. For more information, visit us at emitimaging.com.